All right, looks like we've got most of the people here, so I think we can probably get started. Oh, wrong side. All right, there we go. Took me a couple minutes there to get going. So in terms of the agenda, typical agenda, I'll go through a bit of a summary of our last meeting, and then we'll get into the bulk of the meeting, which is mainly around chemistry, as has been the last few meetings. So I'll turn it over to the UHN team. We'll talk about the some work from Enamine and all the assay data that has come through, and then some next steps, which are some modeling and decisions on compounds to move forward into next set of assays, and then finish off with some additional business, which one of them will be introducing a new collaborator, Max Zub. So at the end, we'll have him introduce himself. Since the last meeting, obviously we had the Christmas break, so that slowed things down a little bit. But we have managed to test over 40 compounds since the last meeting. So 22 of those analogs come from the main series. Those are modifications on the C ring. However, the results there were, I would say, underwhelming. Only 5 of those compounds progressed to dose response. So as a group, we've taken a pause on some of the analogs that we had proposed to make in that series. Essentially, because what we've seen is there's not a lot of wiggle room there, so that pocket is quite tight. So moving forward, we're only going to focus on small modifications of that ring system in that region. So the UHN team has come up with some ideas just to basically make small modifications on what is that thiophene ring in that C portion. As well, I've also had Conrad do some modeling on some proposed small modifications in that system. So just to reiterate, we've paused on making anything that would be bicyclic or large in that region and just focusing on small modifications. For the backup series, we actually had some very interesting results from the variational modeling exercise. So as you recall, was really just a hit finding exercise, and they identified a number of molecules of which Enamine was able to make and deliver 22 of those compounds. And one compound actually came out of that exercise as a potential hit. So it is a micromolar compound, but I think it's great that we been able to potentially identify a hit from a completely different chemical series. So we'll get into that. Um, and then basically taking the SAR as a whole, the team's tested just over 60 compounds. We have 8 that are approximately 1 micromolar or less. So we're proposing to advance these into additional testing. That would be selectivity and cell-based testing. So now I'll turn it over to Frank to give an update on some recent chemistry results from the UHN team.
Great. Thanks, Pete. Yeah, so I'll just go over some of the results or summary of some of our SAR over the past month and a half. This is largely what Pete has already summarized just a few minutes ago, just with structure that everyone can see here. So on our last batch of compounds, we submitted 22 compounds for testing. They were first screened at 10 micromolar, and then basically any of those that have decent, if not good, inhibition at 10 micromolar were then progressed to dose response. Largely, of the majority of 22 compounds did not have good inhibition at 10 micromolar, but these 4 compounds did. And as you can see, they were progressed to dose response. So the best compound here is 2224 with an IC50 of 1.2. And obviously everything was sorry, SGC 6870 was used as a positive control for all of this as well. So from that, as Pete mentioned, we're taking a pause on other kind of C ring analogs. We're focusing on, if you look at 2224, we're focusing on 5-membered ring heterocycles in that C region. So in the meantime, we did synthesize some new analogs. So at the bottom there, you can see 3 newly synthesized analogs that we've completed. And so we're just bringing through some more analogs just to give a bigger batch of compounds to submit for the next round of testing. Next slide, please. Great, thanks. So this is a summary of the best compounds so far to date. As Pete mentioned, you can see our top 4 compounds so far, largely all below 1 micromolar, and they're all derivatives of the thiophene analog. So we're going to explore that a little bit more, where we've proposed some minor modifications around the thiophene ring, as well as varying up the thiophene ring with other rings such as furans, pyrazoles, and then basically doing match pair analysis with those minor modifications on that heterocyclic ring as well. So we've proposed some compounds. We've purchased some reagents in. So synthesis is ongoing based on these new kind of proposed analogs. And so hopefully we'll have new compounds ready for testing soon. Any questions? That's all from my side.
Okay, I do have questions. So these 2 compounds down here at the bottom, they're actually modifying the A region.
Right.
So that'll be interesting. But I do recall from the modeling that extension off of this position actually looks like a better vector to access solvent. So perhaps you can kind of give an update not only on the status of those analogs, but anything that's being done on this aromatic ring down here. And I know that there's been some work done in the background on trying to make that connection asymmetrically. So if you could summarize that, that would be great.
Yeah, sure. So the pyrazine ring location that is coming along, I think, pretty well. So we'll hopefully add on to these 3 analogs. So we'll have a good indication of which one probably will behave better. So that's coming down the pipeline. In terms of the A ring and B ring, both regions, we're still trying to work out the kinks in the chemistry in the hopes of having diversification later on to make our lives a little bit easier if we want to explore both of those regions. As well as the asymmetric stuff is still ongoing. It's not going particularly fruitful, I guess. But we're still putting in some effort in that department as well.
Yeah, I believe the bearing, some of the bearing analogs were kind of promising from a modeling point of view. While C-ring modification, we didn't see anything that was really predicted to be much better than what we had. So yeah, if there is any way to unlock this chemistry around the bearing, that would be fantastic. Yeah, maybe it's not possible. I don't know.
So we did order 3 building blocks based on some of those modeling that Conrad had done that might take advantage of an extra hydrogen bond in that region. So we do have plans to make those, so they are in the pipeline.
Right.
Any additional questions for the UHN team? I'm not sure if Brian from Variational is on the line.
Yes. Hey, Pete. Can you hear me?
Yes.
Okay. Apologies, I had audio issues at the beginning of the call, but I'm ready to discuss Variational's work, where we are with Enamine.
Did you want to share, or you can just give a verbal update on
I
the
have
status?
slides.
You
Yeah.
do have slides?
Yes.
Okay.
Go ahead and share. Okay, can everyone see?
Yes.
All right. So briefly, I'll run through operationally what we did at VAI in conjunction with colleagues at Enamine, identifying compounds from real space, the rapid synthesis and testing that was done, the one-hit compound that we jointly identified, and then plans for how to expand on that using the AI techniques, the machine learning model, and those discussions are ongoing. So the primary driver of the process that we used to actually identify compounds within Enamine's real space that had a good chance, we thought, at being PRMT6 binders in the allosteric pocket was active learning. And the way that we build our active learning loops, it's you can think of it somewhat like the active learning as the DMTA cycle in silico. So compounds are designed by the algorithm. They're made virtually, and then they're tested against some baseline models. So you make predictions, but then you also and then that's machine learning. But then you also use physics-based methods to do a more detailed test. So you have this combination of a prediction of a physics-based model outcome and an actual computed, say, docking score. In this case, that lets you modify, improve your model of the physics-based method. So you adjust the weight to make it perform more like the real in silico experiment. And then finally, all of the model weights are updated. That's the kind of analysis cycle. And that feeds back into the design process. So the active learning really lets you build a powerful docking model and dock orders of magnitude more compounds virtually than you would be able to with current limitations on compute, time, and everything like that. So we've seen it very actively, very rapidly arrive at accurate, at least for docking, predictive models, and that's what was employed here, targeting the novel allosteric pocket. And here's the work that everyone we've been previously discussing that had already gone on. Initial compounds were selected for synthesis. Dave Hathaway presented this along with Pete Guzzo previously, but we had selected 33, I think, diverse compounds from the active learning set. Enki identified kind of two key chemotypes we felt, maybe one if depending on how you look at it. Most things branching from a central nitrogen, some of them branching off of the nitrogen itself, some of them of one or two carbon linker intersecting, and generally with three fairly flexible, three substituents in a fairly flexible molecule that in 2D, it's hard to see the overlap with any of the co-crystallized ligands or the existing chemical matter, but you can kind of see how Enki identified an alternative way to access the same pharmacophore by taking advantage of enamine's diverse real space. I was hoping, so Alex Perryman from VAI, the senior computational chemist who did a lot of the work, has joined me. I thought he could take a minute and just show us the overlay in 3D in Mo of the actual compound along with the co-crystallized ligand, so you can visualize this a little better. But let me first finish. Here are the compounds that were identified. We don't need to belabor this too much. There were 33 compounds, 22, so 67% were successfully delivered, synthesized and delivered within 1 month. So this was really rapid turnaround time. Finley Lee completed the assays early January as soon as the compounds were received. So really moving fantastically quickly. The compound results, there was really one key compound that showed some inhibition, 10 micromolar, and it was this compound here, the thiazole, benzthiazole, and then this spiro oxetane containing compound. You can see some static 2D pictures, and in black is the co-crystallized ligand. It's a bit hard to see. You can see some of the overlap, completely different structure, but accessing the same pharmacophore. This was, I think, the probe information, actually. This is our hit information, and 3 months really from when we joined to be able to build the models, test them, perform experiments in silico, and then for synthesis and testing to arrive at an alternative series that now we can start to talk about how to further evaluate, expand the hit series, and identify if there's SAR trends and something that could be an additional series for development as a backup. Nice looking hill slope. Finally, you had noted that there was some precipitation at 200 micromolar, but generally before that, it looks pretty nice, clean. I think we're happy with that. 13 micromolar IC50 for the structure. And so last thing I want to mention, the next steps that we take. We started here with active learning. We arrived here. So where do we go? When you work with Enamine, a fantastic resource that you have is the SAR by catalog. And so there are significant numbers of compounds that can be mined very rapidly existing within the real space, containing one or two, and various combinations of the different R groups off of that central nitrogen. We have a new technique that we think could prove valuable here. It's called a local search in Encke's latent space. So the model has some high dimensional space where it thinks of chemistry, biology, everything together. This molecule represents a single point in that space. You can look around that point and see what else is there. Encke should think that they are similar, hopefully have similar physical properties, et cetera, biological properties as well. And then finally, we have the ability to constrain the scaffold if we want to start to expand beyond just the make-on-demand space, but take advantage of the core that we found there. We can constrain the scaffold at one or two of the positions, allow Encke to modify. You have a lot of levers to control here as well. So there are a number of paths forward to further develop from the hit compound that we saw. That's all the slides that I had. But Alex, if you're available, I think it might be nice to look in 3D at how we, part of the process that we used to actually triage down many Encke compounds into the 33 that were selected. Let me stop sharing.
Nope. Yeah, I'll probably turn my camera off in a minute just for bandwidth issues. But... Alrighty, can you see my screen?
Yes.
Cool. All right. So in this case, with the black background, the crystallized ligand in 6W6D PDB here is showing like the silver color. And then that's kind of showing you the route that's kind of more exposed to solvent, although not super exposed. So in this case, I did build in the structure of the missing loop using a combination of Mohs homology modeling tools and the AlphaFold model. So these candidates were produced by glide-based active learning in Encke. So there wasn't a specific objective. There were just constraints on like the CNN scores and affinities that evaluate poses and have the deep learning model for rescoring things, as well as the glide score. Here I'll make the crystallized ligand a little less busy, and let's turn off the ribbons for a second. And let's go. Oh yeah. So basically, when I'm doing the visual inspection, I allow some of the side chains to be flexible, minimize it with the ligand, and then minimize the whole system with some restraints. And what we end up with here is some nice electrostatics with that leucine. We're getting a nice T-stacking with the tyrosine. Able to pick up a hydrogen bond with the backbone NH there. Similarly, with this thiazole, we're getting also a nice backbone NH hydrogen bond, as well as some nice T-stacking with that phenylalanine. So it fits fairly well with the crystallized ligand in terms of where it's placing those aromatic systems. And then over here, it's a little funkier with that saturated kind of spiro system. In this case, when I was modeling it, I tried to get some water molecules to do like bridging, but it wasn't really stable during the minimization for this. One other source of potential issue is Schrodinger's ligprep predicts that this is going to be a basic nitrogen. In this case, it wasn't doing anything, and Mo predicts for this compound that it should be neutral. So I fixed the charges before I did the minimizations. But here, it does, for at least two of the three substituents, it does pack nicely, although slightly different angle. And then over here, there's should be room for improvement as well as maybe some room to further grow on this ring. Any specific questions? Or
I was just wondering if you could put the diazepinone back up on over top of that, please.
That'd be this one.
Yeah, I think.
In this case.
Okay, yeah. Just kind of thinking back to some of the SAR we've seen basically on that bromothiophene. Yeah. So it would suggest there isn't really a whole lot of room up off of that ring as well. So I think just kind of reinforces why our thought there.
Yeah, it would have to be something that's not as big and fat as a bromine, probably, if we're growing off the bicyclic.
Yeah.
Oh, and so now I'm showing the, yeah, that's with the interaction shown for that. Let's go back to this guy. But yeah, there is potentially room to grow, although if we do a surface, let's go to whole protein. We'll go lipophilicity. So green is greasy, purple's polar. So there's, so this is a model of the loop that's missing in the crystal structure. So there's not a lot of space. And this also isn't my favorite model of the full domain, but it's the one that happened to do a little better in some benchmarking studies with decoys and your active compounds. So there's maybe some room, and there is some flexibility for the whole loop as well as the side chains around there. So maybe, or maybe you'll have to use like an alkyne or some extension to get through that little choke point and then be able to explore on the surface of the pocket.
Yeah, that was the idea that we had with that piperazine modification that Frank showed earlier. And I think that position on, yeah, that's the other position, right? So,
yep. And so that's the vector to get out to solvent. Yep.
Yeah. And that makes sense. Very nice.
Cool.
So, just ask Dave and Brian, what are your thoughts on the next, on the follow-on for this, from your perspective?
Well, I guess first I'd like to see some of the other candidates from the other active learning experiment, if we can get those synthesized as well, and hopefully maybe the benzodiazepine-like ones. But the local search is very new, and I've been very encouraged by the results I'm seeing in a different project. So that could be interesting to explore, as well as scaffold constraints where we keep 1 or 2 Rs and let it just kind of explore the rest to try to fill in the pocket.
And we actually have a coordination meeting tomorrow. To discuss what's
Okay.
Yeah, I was going to mention the same thing, Brian. Just that, as Alex was saying, right, it'd be interesting to get some of that feedback and be able to implement that into some of more of our modeling, but then identify proposed paths forward, whether it's second round of sort of hit finding, right, modeling or making modifications to our modeling strategies, or focusing on more of the Enki guided optimization that Alex has developed and shown a little bit around the current scaffold and stuff, right? Or do we simply just do more similarity analog expansion around the current hits, right? So we have a meeting to discuss that a little bit further tomorrow and determine next steps.
Cool. Matthew, you had a question?
Very quickly. Well, first, congrats. It's for finding this new scaffold so fast. I have a question maybe for Rachel, if she's here. I was wondering whether there was some structural biology resource to try to solve the crystal structure of this compound. I guess it would be nice to either cement the binding pose or adjust or refine it.
That was going to be my suggestion as well. I'm not sure who in the lab we would ask to do that, but we can. I think it's definitely worthwhile, so that at least we're confident in the binding pose. So, leave that with me. I'll try and figure it out, and we'll go from there.
Cool. Oh, very cool. Any additional questions for the variational team and Enamine team? All right. Oh, that's really cool.
Thank you.
Yeah, thanks. We're excited. And we've got, I think, a number of different paths forward. So, Matthew.
So next on the agenda was Feng-Ling going over some of the assay results. Um, I, I know that Brian already presented this. Feng-Ling, I don't know if you have any additional thing you would like to.
Yes, I can share my on the screen.
Oh, you want to share your screen? Okay.
Yeah. You see there?
Yes.
So Frank and Brian and Dave just shared the compound structure information. Here is the biochemical assay data using the radioactivity-based assay. Recently, we screened 44 compounds and the follow-up IC50 determination. The batch one is 22 compounds from Enamine, and just Brian introduced the structure, the compound structure. These compounds have a different structure backbone from that SGC probe. So we just screen these compounds at 10 and 50 micromolar, and got one compound, the top one, is active. So, and that the IC50 is a potency around 10 micromolar. So this result is very promising, and because this first so small the group of the compound, we got one hit. The batch two is another 22 compounds from Frank. This compound shared a similar structure backbone with the SGC probe. And after screening PMT6 at 10 micromolar in duplicate, we got the 4 hits. And the most potent hit, the top one, IC50 around 1 micromolar. So yeah, this is all the assay data for these 44 compounds. Any question?
Okay. Thank you, Feng-Ling. If you want to stop sharing yours, then I can move on to. Yeah. Okay. Thank you.
Stop.
Okay, so under the next slide. So as I mentioned at the beginning, I'm having Conrad look at some modeling on this C portion of the ring. And essentially what I've asked him to do is remove this carbonyl group and directly link the 5-membered ring or 6-membered ring to the diazepinone. So there are a number of commercially available building blocks that he can look at to screen this. So I think this is just another exercise of looking at potential modifications here that aren't really large, that are because certainly what we've seen is there isn't a lot of room here. So this is in progress, and when those results are available, we can share those with the group. So just to kind of make some decisions on.
Hi,
Sure.
Peter. So I had a question with this, although is to come to you, but maybe as we're all together, we can look at it. So that carbonyl, which we're about removing and then having the ring directly linked to the nitrogen, it was somehow involved in hydrogen bonding.
Mm-hmm.
So I don't know if actually us removing it completely, and then I think we might end up losing that hydrogen bond in
So
most cases.
yeah, I recognize that, but oftentimes you can actually have a bioisosteer with these carbonyl groups with a hetero aromatic. So this is why I've had, these kind of, you can walk around the ring with different heteroatoms that, potentially could still form a hydrogen bond with that background. So if we don't see it from your exercise, then, certainly we don't need to spend time making those molecules. But if we do pick up an extra interaction with any of the things that come out of this screen, then it could be worth pursuing. So yeah, fair point that we recognize that we do have that hydrogen bond, but I think there is a potential to find another analog or bioisosteer that could uh, hydrogen bond to that backbone.
Agreed.
Okay. Yeah, so these are basically the list of compounds that are the most potent. Again, you can see that there is really just a small modification off of these mostly 5-membered rings that we're seeing. And I would suggest that we focus on these 8 compounds that are essentially around a micromolar or less, and move those forwards into methyltransferase selectivity and cell-based assay. And, from there, I think we can look at potentially then chirally separating the active enantiomer out of, whichever ones are the most promising from that exercise to move forward into additional testing. Does anyone have any comments or thoughts on that or agreement? And basically, I'll turn it over to the SGC team. Do you have the bandwidth to run those assays?
Yep, now is a great time to run assays. So yeah, the more the merrier on the compound front.
All right, good. Okay, so we'll move forward with those 8 compounds into additional testing. All right, so now on to some other business. I think I can stop sharing now. I think I'd like to start running these meetings a little bit more frequently, potentially monthly. For the next meeting, I think probably about 6 weeks, so early March. And one of the reasons for that is the law students at the University of Toronto will have completed their IP search on this chemical series by then, so we can have them present in early March. So what I can do is, if this time works, we can pick a time in early March on a Wednesday at 10 o'clock. If it doesn't work, so that'll probably be the second week of March. If it doesn't work, we can try to maybe have a Google Doodle poll to find a suitable time. But essentially, I think moving forward after March, I'd like to keep going with monthly meetings if everyone's comfortable with that. And so next on the agenda, I want to introduce Max Zhu. So he actually contacted me, and interestingly enough, we live in the same hometown, and he is interested in our program. So I'll turn it over to him, and he can introduce himself and describe why he's interested in our program, and two, he's actually done some preliminary business development work for us, which is fantastic. He put our program in front of some groups at the recent JP Morgan investor conference in San Francisco. So, Max, please take it away.
All right, thanks, Peter. Can you hear me well?
Yes.
All right. So Max here, and I live in Oakville, and the reason I'm here, let me see if I can share my screen. The reason I'm here is because my younger son's physics teacher, and he was diagnosed with the SBMA April last year. And so, you know, I don't have to say more. And so I'm here. Let me just share a little bit. Let's see. Can you see the screen?
Yes.
Well, this is my son drew the picture of his teacher during class. So not very good. Well, we love him very much, and he's an excellent teacher. And my son loved that too. Although my son is not very good at physics, but you know, they have fun together. Well, personally, I'm in the pharmaceutical industry, and I have a PhD in pharmaceutical science from University of Georgia back in 2001. And after that, I worked for a number of companies, including Johnson & Johnson, and a number of big pharma, small biotech, as well as contract manufacturing. So I'm on the industry side, and I have done the drug discovery, formulation, computational chemistry, manufacturing, formulation, QC, QA, and finance, BD, clinical trial. I have done many different things, and so I just happen, you know, to be here, and I think I can probably help in different ways or from different angles on this project. Last week, I was in San Francisco, attend JP Morgan as part of my own work. And since I know people in there, I attend the conference every year. I approach a number of potential investors, and very surprisingly, and while the person from investment firm is called Trillium Medical Alliance, and he agreed to meet me in San Francisco, and we had a great conversation. And he introduced, well, he provided a number of additional network, and he also very much interested in Agora's other program. So I think Max and Pete from Agora, and we're gonna have a follow-up meeting with this person, and as well as his network. Also, in the JP Morgan conference, I also met the Variational AI team, and Brian probably know. I met he from Variational. I met Handel, right? Yeah, Handel. Yeah, and Valentine. And so we had a very good conversation, and then we're probably going to discuss some additional collaboration from my own work, and which we're going to have another meeting. I also met another company in JP Morgan, and who's also working on SBMA, And they are using approach of a small molecule mRNA degrader, and still in the very early stage. And they plan to attend the kidney disease conference at end of February. So we're going to meet him. Oh, by the way, I will attend that conference as well. Hopefully, I will have additional fundraising update. So that's it. Thank you. Thank you very much.
Thanks, Max. I think this, is amazing for you to reach out to us, and I think that just kind of shows what our collaboration can do once we get the word out, that if you find like-minded individuals like yourself that are interested, it just adds so much to the program. So, I'm really appreciative of you reaching out and actually doing, this work on our behalf and putting our program into some people that might be interested in talking to us further. So thank you.
You're welcome.
Any comments or questions for Max? Rachel does.
Hey, Max. Thank you so much for sharing that story. That was really fantastic to hear and very inspiring. I'll also be at the meeting in Orlando, so I hope that we can connect in person there. If we don't connect beforehand, it'd be really great to meet with you and talk further about this project and other interests. Yeah, thank you.
Definitely. Thank you.
Hey, that was all I had on the agenda for today. Does anyone have any other comments or questions or other business they want to discuss before I let everyone go?
Just from the enamine variational side, as I mentioned, we have a alignment meeting tomorrow. So, yeah, as we align between our organizations and then internally for approval steps, we'll probably get back to you, Pete, and let you know sort of what our plan is for next steps.
Perfect. Sounds great. All right, everyone. I'll like I said, I'll try to schedule a meeting for early March, and if there's anything that comes out before then, I'll certainly share that with the team. I'll put together summary as usual and send that out to everyone sometime this week. Okay. Thanks, everyone.
Bye, everyone.
All right. We'll talk to you soon. Bye.
Bye.